So I'm going to talk a little bit about vision today, but really only peripherally. And most of the talk is devoted to questions concerning behavior and genetics, which is an area that my lab has been focusing a lot on in the past couple years. And to introduce the subject of behavioral and behavioral genetics, I thought I'd just mention a fascinating example of invertebrate behavior, which is the ability of a spider, and it's an innate behavior in this case, to weave a web of very specific structure and architectural elements. And so this is the web of an orb spider, and as I guess many of you are aware, I don't know how many people have actually studied spiders in any depth. Not me, but I brought it up. But it has been studied a bit, not a lot actually. So you can break a web into its component parts, and so you can see the main sort of frame in red of the web, and then these elements, tangential elements, like the spokes on a wheel that provide a lot of the structural integrity to the web. And then there's the circumferential weave that the spider puts out that is effective in trapping its prey. Now, any particular species of spider actually will generate a web of a structure of its species. In other words, it varies from one species to the next, and in particular along specific lines of these architectural elements I mentioned. And so, for one thing, if one watches, and unfortunately I didn't manage to get a movie of a spider making a web, but if one looks at a tracing of a spider's movements, so shown here in gray are the tracing from A, B, C a long time, and the spider moves along here, secreting the thread behind it, and moves along in a stereotyped way. Let's see, it's going here and pulling a thread there, and then it makes a downward trajectory pulling a thread behind it, etc., till it provides the framework and some of the spokes. And then once it has a nice strong structure in place, it starts putting these circumferential weaves in, and that's where it then traps the prey. Now, it does this in a very stereotyped way, so every spider of a particular species is going to do this in this particular way so that the web comes out according to its species-specific pattern. And so if one looks at different species of spiders and their webs, you can define how they're different by the number of these tangential elements, by the number of bridges that connect them, and then by the density with which they weave their circumferential thread, etc. And there's been some study of how this varies amongst populations of closely related spiders. And a good place, and in many places, many species studies are done in Hawaii because it's an isolated group of islands in which a small number of founder organisms then generated a lot of diversity, and that is presumably true in the case of the Tetrachnatha species of orb spiders. And if one goes to the different islands and looks at different species, you can see how, it's not easy to see here, but you can see how the webs vary. For example, here the density of the circumferential elements compared in this species here, etc. So I'm not going to try to describe all these things, but just to note that each species on a different island is making somewhat different web according to its own internal program of movements as it moves along and generates the thing, right? It seems to know innately how to do this. And the question is, how do genes vary in the genome in order to produce this diversity? And this is, of course, diversity that occurs through evolution. Most people who study evolution really focus on morphology, and they say, okay, well, this animal and that one, they look, a little different in this and that way, and they define certain characteristics. Even or more in the more modern form, they define characteristics in terms of DNA sequence homology. But there's not a lot of study of how behaviors vary in evolution, and yet the variation of behavior in evolution is really one of the principal things that makes animals of a different species different, in particular when it relates to issues such as mating. So for example, mate choice. Of course, an animal needs to choose a mate of the same species. It needs to recognize and choose that. And so one of the principal things that happens when you get a new species is the identification of a mate of the same species. So behavior plays into speciation in a very important way. In other words, right at the outset when you have animals capable of complicated behaviors. If one looks at the different islands in the Hawaiian chain and looks at where this different species that form the different types of web are, what you find is that actually spiders that make very similar webs are not the most closely related evolutionarily. That, for example, these two here, which I'm showing, the light blue circled ones, make a very similar web, but by other criteria, they're actually very far apart in evolution. And so this would be what's called an example of convergent evolution. That is, they separated long ago, but somehow they came out in the end producing the very same. Of course, it's more typically applied to morphology, but in this case, it's applied to behavior. And that seems to be the general case. In other words, when you see a web of the same type, it's often not in the most closely related species, but one that's actually far away. So it's them arriving at the same behavior, probably in a different environment for different reasons. For example, the size of the prey dictates the density of the circumferential elements. But the main question for a geneticist is, what is it that defines these differences as they occur in evolution? Is it a single gene that mutates, that somehow dictates a whole range of behavioral movements that the animal makes? Or is it really a complicated process of changing many genes that makes them have a different innate behavior as a species? And that's something we don't know the answer to, and it's really an area that's going to become a focus of a lot of work, I think, in the next 20 years or so. So any questions? So, yeah.
The numbers, what do the numbers mean? I'm just curious. If that's too crazy, you don't have to get into it.
Yeah, actually, I forget what it was. See, I don't work on that. So I know it's distance in some form, but I'm not sure exactly what the units are of distance here. So if one looks at this sort of in a priori sense, and in the more modern era, because I'm going to talk about model organisms, I'm going to talk about Drosophila, and how many people are acquainted with research in Drosophila? So, because it's one of those things you can bring into a high school, right? And you can set up experiments, and it's actually cheap, which is good. Okay. How does that work? Is it so? I know there's a place that you get them in the mail, right? There's a Carolina, right? Okay. So Carolina sends various things, including instant food and this sort of stuff, right? Okay. Or you go and get bananas or something. Okay.
Or take over.
Right. Well, we have solutions to all those things. You can consult me on that. So anyway, so in the case of behaviors, well, I guess you all know what behavior is, at least when we talk about from the perspective of ethology in animals. That's various stereotyped behaviors, such as courtship, predation, nurturing, nest building is one that's well studied. There's migratory behavior, and of course on the other side of predation is predator avoidance, web building, etc. And many of these behaviors are innate. But even behaviors that are considered innate may be somewhat plastic in terms of experience. And of course, it's only when you get to very advanced animals, such as humans or other mammals, and I don't want to leave out any important, whatever, reptiles, I don't know. I don't think they're too plastic. But even a simple animal like a fruit fly shows plasticity in its behavior. And many of the plasticities it shows are in the modification of innate behaviors, which I'll get to in a bit. So the question that was posed back in the 1970s by a pioneer in behavioral genetics, especially for those who work with fruit flies, was do genes specifically influence behavior? And I have to sort of distinguish that from the general idea that, well, that many genes influence behavior, because if an animal is tired because it doesn't make a proper supply of ATP, or it has, you know, musculature defects or that sort of thing, then of course it's going to have differences from the average in its behavior. But the question that was posed is, are there like specific super genes that really function in the genome to dictate behavior in some way, which are sites of mutation when you mutate them this way or that way, that could cause the animal to change the structure of its web or something of that sort? And that's what many of the geneticists were after beginning in the 1970s, looking for such genes. Now, things turned out to be a lot more complicated than that, because when they started looking for these genes, what they found was that, well, they got all kinds of mutations that changed behavior, but they changed behavior for these other reasons I mentioned. Their mitochondria were not functioning properly, and so the animals didn't move, but that's because they didn't have energy. So it's not a deep insight into behavior at all. And so many of the people who started off to study behavior by genetics in this way ended up studying something else, like how the nervous system develops, because what they got is many mutations that affect how the nervous system gets wired up. Okay? So it didn't lead to a lot of great insight into the direct relationship of genes to behavior. But still, there's a feeling, well, it's out there somehow, especially if you look at genes that influence evolutionary change in behavior. So it's basically an unanswered question, which is why there's a question mark here. Okay, so these are all points that I've raised, and I think that's sort of visible. But it's okay. So what about a behavior like aggression? And if you consider aggression, it's something that you see between animals of many different species, including even fruit flies. And so just to show you that even fruit flies can fight, and this is an experiment you can do in a high school lab, I presume. So here we have boxing between fruit flies, and I want to credit Ed Kravitz over at the medical school for taking these. So this is a female, and these are a pair of males, and what they do, and this is really truly stereotyped aggressive behavior, is fight in the presence of a female. Yes. So, well, let's see how much time we have. We can watch the whole fight if you like. Right, so to an ethologist, they break the behavior into many different components. And so, for example, sticking the wings up like this is a threat. It's basic, just like a cat, you know, makes me look bigger, right? And, yes, okay. Tussling. So all you really need to do this, okay, the female flew away, which again, yeah, this often happens. And so, but this is actually, so I know you can find out how to do this, and you can actually, you need to videotape it or something, but basically you just need a dish like this and a pair of males and a female, and you can observe this behavior. I could point you in the direction of publications. So, as I mentioned, back in the early 70s, the notion that genes determined specific behaviors was really attributed to its champion and one of those progenitors in fruit fly research that many of us who are in it now look to as their inspiration for getting involved. And in fact, I can recall when I was a grad student reading a Scientific American article by Seymour Benzer and thinking, gee, this is really an area that would be nice to work in somewhere down the road. So he published a paper in the Journal of the American Medical Association, no less. I mean, I don't know how it got in there because he works on fruit flies. But it was from gene to behavior, and discussed the relationship of genes and behavior. And in particular, which I'll show you in a second, a lot of mutants in fruit fly behavior that in very many different aspects of fruit fly behavior. But he was also the first to show that you could train fruit flies to do things. And so that's what this is referring to, conditioned behavior of Drosophila melanogaster. And he isolated a mutant that made fruit flies deficient in learning. And also, if any of you have heard about the control of the circadian clock, and so how many people have heard of circadian clock genes? And that came, the first gene of the clock circuitry was identified in his lab, and it's known as period for periodicity. And now there's an entire very complicated mechanism of circadian change in the levels of certain proteins that underlie, and of course it's all conserved to humans too, we know that now too. So, and if you want to read more about him, there's a biography by a well-known author, Jonathan Weiner, Time, Love, and Memory, and it's all about his work and his life. I don't know if that's beach reading, but it's... So amongst the classes of mutants that were isolated are ones that affect locomotion. So the animals were sluggish and hyperkinetic, which is described as living fast and dying young. And then we have animals which have problems in their response to stress. So if you bump or bang these mutants called easily shocked, they fall on their back and exude fluid or eggs and lapse into a coma. But then they recover over time. So it's some sort of epileptic type dysfunction. It's actually, these are things that are of interest to people who want to study disease, is to understand what's wrong in these different mutants. Shaker is a mutation that actually led to identification of the first channel in neuronal membranes that lets potassium out during action potential. And it was identified mainly because when they etherized the flies, which we don't do anymore. I don't know if you use ether, but we use CO2, right? Because it's not whatever, explosive. And freaked out. Okay, was going too slow. Spinster is females reject males. Fruitless, males court males. And that's turned out to be a very, even now, a really detailed study into the mechanism of a particular protein that's expressed in a few neurons in the nervous system. And a different form of it is expressed in males and in females. So it's under the control of the sex hierarchy. And then it, in some manner, affects how the males and females behave, makes them behave differently, which people don't know. Now I'm going to focus here on a particular example, which is Phototaxis and natural strains of fruit flies. And I guess anybody who's held a bottle of fruit flies up to the window knows this, that they move towards light. And so if we put them in a tube, also a very easy experiment to fool around with in a high school lab, I think. I mean, it doesn't require anything more than a tube and a light. And the flies respond to this with positive phototaxis, usually moving towards the light. Some don't. And it's actually an interesting feature of the animal that not all animals respond in a given situation. It's as though they're doing something else with their minds at a particular time. So for example, if you took this animal, which for whatever reason didn't go to the light, and took the animals like this, and now you do another experiment with those animals, you say, okay, well, now do they respond the second time? You might, I think, not find that there's actually a lower frequency of response in the, this non-responder population. So they're off dreaming, daydreaming and stuff. Now, Benzer actually used this phenomenon and his background in physics and chemistry. So I don't know how many people here have a physics or chemistry background, but if you've done serial extraction, he invented the equivalent, with the organic and inorganic phase, or organic and aqueous phase, extraction of fruit flies with serial trials to purify a population of fruit flies that didn't have positive phototaxis. And so the way this works is you start off with 2 tubes, and you have a population of flies, these little dots here, and you turn on a light in the empty tube right up here. Now the flies will phototax. Now, you can imagine that this population of animals are all animals that have different mutations, and some of them may have mutations that affect their phototactic behavior. And so after that light trial and the phototaxis, there's a probability that a fly will have responded to the light, which we'll call P, and the flies that don't respond, 1 minus P. And so now you take, of course, these might be enriched for animals that don't see light or respond to light that don't have positive phototaxis. You take these 2 tubes, you put this one here, you put this one there, and now you repeat the thing. You take 2 fresh tubes here, and now you turn on the light up there. Okay, now after that trial, you'll have, how many people know their statistics here? So what's the probability, how many flies will be up here? Yes, teachers? Yes. I'm getting you back. Okay. Okay, well, it's going to be P 1 minus P, because it's 1 minus P is the starting population, and the probability of response is P, so it's P times 1 minus P, and etc. So it's actually all up over here. So these tubes are now, oh, this is after the trial. Huh, you could have looked right there, see. Okay. So P1 minus P. Those that responded with positive phototaxis twice are obviously P squared, okay, etc. And so the ones that now have failed in 2 trials are 1 minus P squared, and there's fewer of them. And they're even more enriched for animals that may have mutations in genes that affect positive phototaxis. And that's how he went about isolating mutations, and a lot of interesting genes were identified this way. Now, you actually, at the beginning, there was a discussion. Oh, you brought up the apparatus in the high school workshop protocol that we devised. And so we made something very similar to this, but that does it without a whole bunch of tubes, and is far less complicated. And so in this apparatus, which is also, it's not hard to build. It's just an assembly of diodes, blue diodes, and we put green ones in here, but you can leave them out. Let's say you want to have the animals make a choice between light and dark. So just consider blue to be the light on. If, and there's a hole at the bottom here, and this, we normally have a cover on here so you can't see. These are tunnels, okay? So the flies go in here, and of course you also maybe know about flies is that they tend to walk upwards. In other words, they have negative geotaxis, so they will walk up. And so you put them in this hole in the bottom here, and they walk up the tube, and now they come to this choice point. And so they have a probability of P of walking towards the light here, or a probability of 1 minus P of walking towards, well, let's say that's dark. And then they get to the next choice point, and they make another choice. Yes?
Is there any way that you sort of, I don't know, I guess averaged for like group behavior?
We looked at this because, of course, as soon as we built the thing, we wanted to know, do the flies kind of all want to hang out together. Yeah, and one phenomenon that's not unusual to see is that if a female walks up here, the males will follow. Okay, then they pay more attention. So there's potentially, or if an animal that has food odors on it walks up there, and then they'll follow up along a path by smell, not sense of light. So we have to look at the distribution of animals and across the entire apparatus, and what we tend to find is, for one thing, that it's a binomial distribution, so we don't see a lot of clumping. In other words, the number that you find coming out of each of these holes at the end is predicted by independent probabilities of the choice at each of the nodes when you count them. But even if we put single flies and we watch the behavior of single flies, they seem to make this choice independently. They're not in groups. So we put a clear plastic cover over it and then we look or something. So we think it's pretty independent. But we do wash it out once in a while in case there's like smells or something. So, all right. So basically the same kind of statistics go on here. So for example, if an animal goes p here towards the light and p there and then p there, it's p cubed is the probability. And meanwhile, this, and that exit port, and we put a tube up there, is 1 minus p cubed is the probability. And of course, those would be the phototaxis, the positive phototaxis defective animals. Or even, so if you start off with mutagenized animals, which is probably not something you do in the high school, right? Work with mutagens, right? Oh, okay. But you might get out of this tube just the students. Okay, whatever they're doing. So those animals might be enriched for mutants in that tube that actually don't like light. For example, if you hit a gene that changed the interest of an animal in light, made it run away from light, it might end up there. So we've made a somewhat more advanced apparatus. It's not worth going into. But we've also managed to train flies. So this is where it gets into this question of, well, of course, innate behavior, which I mentioned, which is to go towards light. But there's also some plasticity involved. And we're able to change their behavior by training them. And the way we train them, and this is also part of the workshop that was mentioned, is to make use of a PCR machine. And the fruit flies are put into a PCR tube that has a light in the top. And when the temperature in the PCR machine goes up to an uncomfortable temperature for the fruit flies, the light goes on. And when the temperature quickly cools down to something more comfortable, like 16 degrees or 20 degrees, the light goes off. And so they are supposed to, or in this, they're thought to learn an association between some uncomfortable heat, unconditioned stimulus is what it's referred to, and the conditioned stimulus, which is the light. And then we put them in the apparatus and measure their phototactic propensity, in other words, the probability result from each of the node choices. And what we find is that they don't learn to run away from the light, but what they do is they stay put. In other words, they learn light is not something they want to go towards. So fruit flies are capable of doing that. They're not capable of overriding their innate behavior by saying, I want to run away. Yeah?
How long does that behavior last?
It is strong for about 45 minutes, and it begins to decay, and I think they're almost back to normal by about 6 to 8 hours. And that actually has something I'll talk a bit about, which is short-term memory and long-term memory. If I don't go if I'm going at the pace I'm going now, I don't know. But we also did another kind of experiment. This is a more sophisticated type of experiment, and I haven't actually shown you a picture of a fruit fly brain yet, but involves the rather complicated circuitry of the fly brain, so you might not have imagined because it's awfully small. But anatomically, you can break it down into different parts which, by behavioral studies and genetics, have been assigned particular functions. And so, for example, there's a region here called the mushroom body, and I don't know if that's because it looks like a mushroom. And it's found to be a place required for olfactory learning, which I'll get to in a second. Motor control seems to be in this region known as the central complex. And we have the ability to do experiments now where we turn off the function of particular neurons in the brain. In other words, we can say, we want to turn off this neuron and just silence it. And of course, this is a live animal. So this is an animal that's awake and behaving and doing things we want it to do, except that we're changing the ability of a few neurons to function. So actually, it turns out when we knock out a few neurons in here, we can make the animals behave differently towards light, which is to run away from it. Now, I'm wondering now if you, because we did this experiment and we're trying to get it published now, and we don't know if it's going to, what the reviewers of our paper are going to think. But so we did this experiment and it gave us what you might consider, I don't know if it's a counterintuitive result or not. We did our heat-associated training experiment in which we took these flies which run away from light and we put them in the PCR machine and subject them to heat in the presence of light, etc., like I've told you. So what would you expect them to do? I mean, normally, they seem here to be doing the right thing, which is running away from light after, even without training, right? So being that fruit flies are semi-intelligent creatures, what would you expect to happen if you do the training with these animals? Sorry?
They would stay away.
They would just do what they're doing because it's a good idea, right? Is that okay? Yes?
Would the ones who you suspect to have, as you were saying before, like the visual problems, the ones who I guess don't recognize the light, what do you call that?
Yeah.
Like after you went through it, you figured that those were the ones who weren't able to recognize the light. Would those be the ones that still are not moving at all?
Well, they might be ones, yeah, except that if you haven't, most of the time, if you haven't mutagenized and you take those animals, unless they're naturally occurring mutants, or
so you're working with them not in your population.
Not in our population. That's what we're doing now. Yeah, we're not taking mutants now. Yeah. Okay. Any other bets or not bets or whatever? Okay. So the result was that after training, I don't know if I put this here. Oh no, I didn't. Okay. After training, they just stay here, just like the normal animals that didn't have these neurons silenced. So in other words, they do the counterintuitive thing. They do actually kind of a stupid thing, which is instead of doing what was a good idea, they do the thing that seems to be the same learned behavior as the animals that normally go towards light, which in that context, it is a smart behavior, right? Yeah.
Did you reverse it and try and see if you could train them to go to the light since the negative phototaxis is there? Did you then train them to have heat?
We haven't tried that. Yeah, if we turn off the light and heat them and then try to train, no, we haven't tried it, but maybe that's what the reviewers will say when we.
Darkness and the heat.
Yeah, right, darkness and heat, good idea, yeah. Yeah, yes.
Could they be heat loving at all and not light?
Well, usually you pick something that they're just not likely to they have, well, good reason not to like, right? Heat, because it could hurt them. So they probably don't like it, but let me see. This only happens with the train. Yeah, maybe you're right. I can see what you're saying, right? Yeah, they're learning to stay put because they like the heat. Yeah. Well, that's another thought. Okay. We could do a test for that. Like we could give them a choice between a hot place and a cold place, and that would be sort of a control. So yeah.
Especially if that hot was the same temperature as your PCR.
Right. That's true. Yeah.
Just going back, these are not mutants, you said they're wild type, and did I mean
Yeah.
that you were actually creating lesions in the brain or something to make them
Right. Just specific neurons are expressing a protein that shuts them down, so they can't fire. So they can't be part of a circuit. So we're altering the circuit in some way, but we haven't explored yet. But we're looking at now what do those neurons do? because they're interesting neurons to us because it's going to potentially tell us something about the circuit for the behavior, and which is something that a lot of people are focusing their attention on now, is even though it's a very small brain to work with, which makes it difficult, it's still possible because with fruit flies there's a lot of powerful genetic techniques to do things like knock out specific neurons or hyperactivate them and really begin to pick apart circuits in ways that might even be even much harder in a very complicated brain like that of a mouse, for example.
You're saying the Drosophila brain is understood well enough so that you can specifically create, or mutants already exist, that eliminate some of the components of different neural pathways?
Well,
Is that what you're saying?
the methods really aren't to eliminate the neurons. The neurons are there, but what we do, and I don't know, probably, so like you haven't heard of the system called GAL4 UAS system. So it's all involves transgenic animals, and there's a protein called GAL4, which is a transcriptional activator. And then it can be, you can generate collections of animals that have the expression of that protein in different neurons. And so we have them. We have like hundreds of different strains, and they all have the expression of that protein in different neurons, not the same neurons. So we take things from that library and we cross one of those to a strain that carries the promoter that transcription factor works on upstream of a gene that will shut down the neuron. So we cross those two things together, and now you get an animal whose brain is expressing in a particular number of neurons this gene downstream of the promoter, which is interfering with the secretion of neurotransmitter. So we shut them down. And we could sort of go through our library of different strains, and we can say, oh, we shut down these neurons, or those neurons, or those neurons, and what happens, and how's their behavior affected, which is really the approach we really take. You know, as opposed to saying, gee, I think I'll go, knock out those neurons over there and see what happens. We just sort of do more of a unbiased screen for finding things that affect behavior.
I was just wondering, is there any degree of heat that the light gives off that might be part of the reason as to why they don't go?
Yeah,
I don't know if that's sort of what you're
we concern ourselves, but the diodes don't give out much heat, and we made sure to keep them far enough away from the tube so that we're pretty sure that's not affecting them. So, okay, so I can go on, and I can even skip things. So, well, towards the end of the talk here, I'm going to talk about another class of Benzer's mutants. I mentioned he isolated a mutant called dunce, and other mutants that affected learning or memory, which they, for whatever reason, named after vegetables. So rutabaga, turnip, and I can't quite remember, radish is another one. And these have been studied in the last 15 or 20 years, also in conjunction with studies of the mechanism of memory in other organisms, most notably aplysia, which is a small marine mollusk, for which the Nobel Prize was given out to Eric Kandel a few years ago, for identifying components of the intracellular messengers that are involved in memory in that really simple set of circuits that's there in what's controlling the gill, because when you poke it, the gill is withdrawn. And so he identified cyclic AMP as an intracellular messenger involved in that response and in changing the plasticity of that response, that is. And of course, to make cyclic AMP, you have adenylate cyclase, and to destroy it, you have cyclic AMP phosphodiesterase. So these are all things involved in the metabolism of cyclic AMP. So it turned out that Drosophila mutants defective in short-term memory, like dunce and rutabaga, were defective in the same things that we now think are universally involved in modifying the connections between neurons during learning, at least short-term learning. And I don't know if people have experience, but know that there's different forms of learning, short-term, long-term, right? And I think everyone knows just from their own experience that short-term is something that you might learn and lose very quickly after a single episode of experience, right? Like hearing a telephone number. But long-term learning is something you get after repeating something over and over again. It takes longer to a longer training episode and a particular type of training. And this might actually be useful in high school, I should mention. So when you teach your students something, you should space it every 15 minutes, do it over again. That's what works best for fruit flies. So, but I'll go through this a little bit, how we train fruit flies to dislike a particular odor. And we use an apparatus that is probably a little too complicated to set up in a high school. You need a vacuum pump, and you have a little cup here with an odorant. And so we just use chemicals like octanol or methylcyclohexanol. There's various odors, benzaldehyde that fruit flies don't like. And so when that odor is drawn through a chamber containing the fruit flies by the vacuum, at the same time that occurs, you are giving them electric shocks with a pulsating 90-volt copper grid that's in there. So they're sitting on the grid getting shocks to their feet as they smell that odor. And that takes about a minute. And then you give them another odor, and there's a reason experimentally to do this. But this second odor, odor B, is not coupled with the shock. Now we move over, I think, to the assay. Right. So now in the assay apparatus, we have both of these odors coming from separate sides, and we put the fruit flies in the middle. Now, they don't like either odor to begin with, but we set it up so that normally they're rather, in other words, the strength of this odor and the strength of that odor means that they pretty much split 50-50. They'll come out of the center and they'll go this way or that way, but they'll split 50-50. But let's suppose, and I've shown you, that they've been electric shocked in the presence of odorant A. And so what will then happen is pretty much all, except for a few, I don't know what, they'll go over to the side that has odorant B. In other words, their repulsion from odorant A has increased in intensity because they've experienced a shock with it. And don't want to get into how we quantify that, but we do the experiment with, say, coupling the odorant B with the shock, and then we calculate something called a performance index, which basically tells you how well they learned. Now, if that's done, I should say, show this. If that's done with, say, odor A with a shock for 1 minute and then odor B, and then we do the behavioral assay, and that's it, they acquire short-term memory in that assay that I showed you. And that short-term memory declines very quickly in the course of an hour. So by the end of an hour, they don't remember anything. It's down to nothing. Now, there's a form of memory that I'm not going to talk about, which is called middle-term memory. And then there's long-term memory. So fruit flies have it too, just like people. But to get long-term memory, you can't do the short-term memory paradigm. You have to do the training 10 times over a period of 2 hours. And so you do the odor A with the shock for a minute and the odor B. You give them a sabbatical, and then you repeat it 9 times. And you can assay even 20 hours or even 2 days later. Now, first of all, they don't even have long-term memory up to about 4 hours. So it's something at the end of the training that they acquire slowly. And then once they acquire it, it's very stable. And so this is 24 hours out here, so that this just stays like this for several days. It just never is as good as short-term memory, but it's far more stable. And it's also very similar phenomenon to what you see in more higher animals like mammals. And another feature of long-term memory in such complicated animals is that it requires protein synthesis. And so that's sort of the state of knowledge. In other words, people know that to make a memory stable, you need to make protein, which is, of course, why you have to tell the kids to eat a good breakfast, right? Okay, so you repeat it over every 10 minutes, and they have to eat. And then they make proteins, because in a mouse or a fruit fly, if you block protein synthesis, they can't acquire long-term memory. Now, what's thought to happen in an animal that gets a long-term memory is that new proteins are made at synapses. And it may be some synapses, or it may be a lot of synapses, maybe a few or a lot. Nobody really knows. But the synthesis of those proteins at synapses is thought to change the synapse in a very, or be required to change the synapse in a very stable way. And of course, changing synapses in a stable way is what's thought to change the way a circuit in the brain works. And that's eventually, well, that is what is thought to be a memory, is some combination of changes in a circuit. Yeah?
How critical is the sabbatical period with
It-
respect to the repeats?
It's very critical. So there's another paradigm in training that's known as mass training, and basically you just do it over and over again without that space. And I think the result is that they learn a little bit, but they definitely don't learn as well. So we have to do it this way.
Can't cram for the exam, see? You don't.
Well, that might be short-term memory or middle term. But yeah, I would say that's probably not the way to remember something 2 years later. But I don't, just kidding. Sort of. I'm not a psychologist, right? So yeah.
You said the long-term memory eventually disappears after a few days?
Yes, but of course the fruit flies only live a few weeks. So, in context.
No. My question is, does it take less to reintroduce that to a long-term memory? So say after 3 days,
Yeah.
they lose, the memory's gone. However, after once or two or three retrainings, does it stay
Yeah.
with the long-term memory?
I don't know the answer to that. So I mean, that would be something interesting to try. These are all things, if you manage to figure out a way. Of course, it's not easy to set up this particular type of apparatus, to set it up and get it to work, because it requires shocks given in a certain way and the odor sensing and all of this. It's not an easy experiment to do. But one of the things that strikes me is that, a lot of this stuff is something that if you just build some little gadget, you can do it in your garage. I mean, you can go out and get Octanol or something, right? I don't know, somewhere, but, and
I was wondering if you had done anything like that with a scent that they like versus a scent that they don't like?
I think it's been done. I mean, of course, there's the odor of bananas, that sort of thing, or fruits, and they're attracted to. I think in the field, so people who work in this field of learning, olfactory learning, tend to be very rigid because they all want to adhere to the same paradigm. So they don't explore very much. So I've heard, and I know in early papers that they found that they couldn't get very consistent learning with attractive odors. That they found that when they're balancing animals between two repulsive odors, that's when they could get very strong differential behavior, and so that worked very well.
I wonder if that has anything to do with what's already hardwired into their genes.
Probably. Just like in the Phototaxis case, it's not easy to make them run away from something they're naturally attracted to. So it's a limit on the ability of plasticity to affect and hardwired behavior. Yeah.
Again, understanding that fruit flies only live a short lifetime.
Yeah.
Is there a certain age that you use for these? I mean, is there, we
Always-
talked about a little bit about
Always in the first few days
the first few days.
after they come out of their pupal case, so they are young adults. I think a lot of the reason people do it that way is just to have very standardized conditions. So I don't know the result, if you try how learning decreases with age. But I think experiments like that have been done, and it probably does decrease with age. Sorry to say, but of course, and then an interesting line of research would be how to correct that. So, and it's almost 11 o'clock, and I had a whole other thing to talk about, but.
We have some flexible time, so
Oh, no, that's okay. I'm just kidding. No. Anyway, okay, I could skip some stuff. Okay, well, I will skip stuff. I was going to say that fruit fly brains, though small, have structurally a lot in common with much more complicated large brains like humans. And so, for example, in a mammal, in the cerebellum, you have what's a very elaborate structure, the dendritic tree of a Purkinje neuron. In a fruit fly, you can find neurons that have dendritic trees. That's all the postsynaptic sites there, and then the axon goes down here. They're just much smaller. So the scale, you can see the scale bars, 10 microns across versus 100. So a factor of 10 smaller just in the size of this structure. But it's another thing to think about is, when you have something that's so tiny that you can't see it without a microscope, that, how did nature manage to make animals all these different sizes and yet preserve the structures? And of course, they also preserve a lot of their functional abilities. So let's see, skipping this, skipping that. Okay. So I'm going to talk about a protein called CAM kinase 2, which stands for calcium calmodulin dependent kinase 2, which all that means is that it's a protein. It's a rather fancy looking protein, many subunits arrayed in sort of look like a Ferris wheel, that in the presence of calcium will become an active kinase, which means it's going to stick phosphate on serines and threonines on other proteins. Okay, so this is a kinase, and it's found at synapses, which I misspelled. And also, the RNA for this protein is found at synapses too. And what's known about this, and not from fruit flies until what I'm going to tell you about, but in mammals, mainly with work in mice, is that the protein gets synthesized at synapses when a synapse gets activated in a really quick fashion. So if it goes bing, 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 a firing, then all of a sudden there's new CAM kinase 2 made at that synapse. And the RNA is there too. So what's thought to happen is the CAM kinase 2 is made there, and it might then have some role in modifying that synapse in a way to change it, like I told you, is required for long-term memory. So this might be really one of those proteins whose synthesis is required for long-term memory. And there are many mouse studies in CAM kinase 2 mutants which have memory defects. And in fruit flies, they have the same protein. And they also have a certain behavioral defect, at least in terms of what's been looked at so far, that's related to mating behavior, which I won't get digressed into. So what we did, so here's the gene for CAM kinase 2, so the open reading frame. And then at the end of the mRNA, it has something called a UTR, which stands for untranslated region, because this part of it actually is the protein coding sequence, and then this part is just sitting on the end of it. And what's known from the work in vertebrates is that this part sitting on the end of it has sites on it that are what actually make the mRNA get regulated at the synapse by the activity of the synapse. So there's regulatory sequences here, not protein coding sequences. Okay.
So those regulatory sequences are read after the protein is made?
They're on the RNA. So this is the RNA, and the regulatory sequences sit on the RNA, and they actually control whether the protein gets made or not. Whether it gets made or not. So the ribosome has to get on here. It has to go through and read this. And somehow the tail of the RNA is controlling what happens at that other end of it, because it's not actually like nice and straight like this inside the cell. So we made a form of this, and I think someone earlier talked about using fluorescent proteins. So we put a fluorescent protein coding sequence on here with the same regulatory sites at the end of it. And we can use this now to look at protein synthesis at synapses in the fly brain when they're learning. And this is the experiment that we've done in the past several months. So for example, we'll express this protein in this particular type of neuron here. Now this neuron is a critical neuron in the olfactory system. And so this neuron sends its dendrite into this area here where the axons come from their nose, essentially, their antenna, where they sense odors, come in here and contact the dendrites there. And then it has an axon, and it outputs to this region of the brain I mentioned is involved in memory here and behavior over here. So in other words, this might be where the output says, oh, I don't like that odor, run away or something. But so this is an interneuron, and it basically connects one starting end of the circuit to downstream ends of the circuit. And I can kind of skip over this, but I don't want to get too complicated. But basically what this sequence does is cause there to be synthesis of the fluorescent protein in the dendrites of those neurons. Okay, so now they turn fluorescent green. And there's the axons, so the dendrites here, you can see right there, are all fluorescent. And their axonal endings here are fluorescent. And what we know is we can take the brain out and put it in a culture medium and add nicotine, which, I don't know how many people know what nicotine does, when used in a pharmacological sense, but it activates acetylcholine receptors. So it's a way of inducing neural activity. And when we do that, we induce rapid synthesis of now the fluorescent protein in the dendrites. So we can show that neural activity induces that protein to be synthesized. Well, anyway, let me just make this a lot more simpler, what we're doing, and not get into all the technical details. So this part of the brain here, here's the fly's brain. And this isn't a real image. I cut this out and pasted it on here with the green staining of the fluorescent protein in those dendrites, in the sites where olfactory odor sensing comes into the brain here. And the question we want to ask, this is the question, when the animal learns something via long-term memory, in other words, repetitive training, where is protein synthesized? Is it synthesized all over the place, or is it synthesized at specific synapses that are actually somehow part of changing the circuit in a way that is a specific type of change? And this is a question that really hasn't been addressed in any complicated animal, because it's not easy to do it in a brain that's a lot more complicated than this. It's complicated enough, I can tell you. So we do this odor shock training again, and we do the repetitive space training paradigm. And we take the animals which have learned to hate odorant number A there, odorant A, which in this case could be this chemical called octanol, or another one called methylcyclohexanol, or we just call it MCH. And we look then at where those structures in that region I was pointing at become more fluorescent. So if the animal is trained when you pair the shock with the octanol, we see that certain places become more fluorescent. So this is just a scale where blue is not very fluorescent and red is more fluorescent. So if you took the untrained animal here and you look here versus there in a trained animal, you can see it's become redder. So in other words, there's more protein synthesis in this little area, and ditto for here. Whereas if we train them with this other chemical called MCH, you can see now the result is different, because here and there it's pretty much the same, but here it gets a lot more intense when we use that chemical to train them. Yeah?
To ask you a question about the gene,
Yeah.
did you add the fluorescent gene to the CAM kinase 2?
in the so it's a transgenic animal, and the CAMP kinase 2 gene is still there.
Okay.
So they still have their normal gene, which is presumably doing something. Actually, and we have an antibody to recognize that protein, and we can see the protein itself. We don't have to just go by the fluorescent protein, but we can see the protein itself. So anyway, the basic thing here, and what we're doing, well, we've done a bit of, and we're going to do a lot more in the future, is try to figure out in the brain where new proteins are made when the animal is learning something. And that's pretty much a cutting-edge type project and something that's not done before in any animal. So, so it really, when we start to understand stuff like that, we'll start to understand how a circuit changes to make the animal have a different behavioral response after experience. Now, I could go on for another 15 minutes, but it only gets more complicated. So, and I was going to tell you how RNAs affect learning, which might be nice to know about, but I'm not Fidel Castro here. So. And normally when I lecture in my classes is an hour and a half, though. So, but and if there's any more questions. So. Yeah. Oh yeah. Oh yeah. Okay. Okay. Yeah, I'm getting used to the light now, anyway. So. Yeah.
Is there any difference in the degree of aversion to the two chemicals in the untrained animals? In other words, other than just the localization of where the fluorescence occurs after the training, I don't know, just kind of looking at it, there seems to be more blue, for example, in the MCH than there is in the OCT. So,
okay, you mean going down?
Yeah, are there
Right.
different degrees initially in the level of aversion to the two chemicals that correlate with that?
Well, they're untrained. Pretty much in a population, you find the animals have, of course, they don't all behave the same. So you probably have some, of course, if you do any assay, you find that the animals vary in their behavior. And you'd have to take the ones that, say, showed less aversion. But if you consider it as just a population, like an average behavior. So we set up the experiment in a way in which we give an amount of odor that kind of gives a standardized amount of, repulsion, running away. So let's see. So the untrained ones don't change because they basically have that baseline repulsion. The trained ones have much stronger repulsion, right? So now they're running towards something that they don't like. It's not associated with electric shocks. I'm not sure I'm answering your question, but I think I'm kind of beating around it in some way, but.
I guess I was just curious about whether or not in a particular population that showed aversion to both of those chemicals, just looking at the untrained results up there, if there were differences between the levels.
Oh, I see what you're saying, here versus there. No, that's a very good point. Yeah, that's a good point.
I'll ask the same question, the variable. In other words, just
Yeah.
untrained with that and untrained without the shock. But the
Right.
chemical.
Right, untrained. Well, it's not an easy experiment to do. And because we're dealing with populations of animals and we do many different experiments over time. And so then we get, for example, here, basically this is a way of expressing change, delta F over F, which is change in fluorescence over fluorescence. So all of these experiments that involve, no training, or actually no training is not shown here, but let's say where you haven't paired the shock and the chemical, the odor. They're not paired together. They're just alternated. You can see there is some change in some of these different structures, and some of it seems to be statistically significant. So from all of our repetitions, you look at the error bars, well, this would seem to be statistically significant. But of course, if you look at enough data, you're always going to find something that is off the average enough to not fit the rule. Let's say the rule we're trying to say is, well, there's no change if we don't pair the shuck and the odor together, or we don't train them. There's no change, right? But if you look at enough of these structures, you're going to, on average, probably find, in some cases, even if this is a standard deviation or two, that in a set of experiments, that you're going to find a site in the brain which just happens to have given measurements that are 1 or 2 standard deviations off the average. So the answer is, well, if there's a small change, and even if this says it's statistically significant, we're not absolutely sure that it is. And you see a certain amount of variability between, in experiments between these different sites, which you can probably pick up on here. Like if you look untrained, they don't look the same. Those are different animals, and so we're averaging. And so basically, for us, the main result is that when we train, say with octanol, this is with the shock, or MCH with the shock, the changes, the increases are way off all of, you can sort of separate the signal from the chatter, basically, if you look at this. So we're sort of making the argument here that unless we could do this experiment 100 more times, but believe me, the postdoc who did it would never, it's not an easy experiment to do, because it's dissecting open the head of every animal and then analyzing fluorescence in its brain under a microscope, one at a time. So, and then this is involving hundreds of animals, and so it's not easy. So we take what we see as maybe some of this might be real, some of it might be just noise that we haven't gotten rid of, including like differences you see here. This is pretty similar here and here, but you might say, well, this one here and that one different. I don't know, and but these are individual animals, so they're not averaged. Yeah. Okay, so. Yeah, I was just illustrating this, basically how memory is indicated to us, say for a chemical like octanol, by fluorescence in specific sites in the brain. And what we're thinking is that when you make protein at one site in the brain, that it's part of making a signal that sends the memory perhaps on to other parts of the brain where people know from other experiments that the memory is more stably maintained. So this area over here, we don't think is a place where they keep their memories for many hours, and there's evidence for that that I won't go into. But so we think maybe making protein here is just part of transferring memory to somewhere else. But that's another question we're going to try to address in the future. Well, anyway, I mentioned there were regulatory sites at the end of the CAMP kinase 2 mRNA. And I sort of mentioned it before because when we started this series of experiments, we thought it was because these sites were the binding sites for proteins. But it turns out, and we have now an awful lot of evidence for this, is that it's not the binding site for proteins, but it's the binding site for other RNAs, which are called microRNAs. And I don't know if, by reading the news or whatever, you've heard of RNA interference. And, right, so how many people have heard of RNA interference? So a lot of people have heard of it. So it's really a recently discovered phenomenon where you can introduce an RNA into an animal. So this is from the outside. And of course, this happens a lot of times in the case of RNA viruses, that an RNA comes into a cell from the outside. And the cell knows how to respond to it, or will respond to it, by chopping up that RNA into little pieces, which is all of this biochemical stuff over here. It's taking the RNA and chopping it into little pieces and puts it into a protein complex that then goes around the cell and looks for any mRNA that has the same or very similar sequence. And so it's what's referred to as a surveillance mechanism. In other words, this complex will hold an RNA as sort of its recognition template, go around the cell and find another mRNA, and it will do either of two things. It will chop this up, destroy it, which is a good idea if it's a virus, right? Or it will just silence it so that it can't be turned into protein, which is another thing that can happen. And these things called microRNAs are not RNAs that come into the cell from the outside, but they're actually RNAs that are made in the cell itself to control the expression of its genes. And this has also just been discovered in the last few years. And so it's really an area of research where things haven't been sorted out, but we're beginning to get an idea of what's going on. So, for example, during the development of a fruit fly in the embryo, in the genome, there are a bunch of these microRNAs of different sequence, about 40 or 50 of them, maybe more. And they're expressed in a certain way that actually control whether proteins are getting made or not made. And so if you take them away or mess them up, the animal can't even develop properly. So these things turn out to be all involved in just the normal development and functioning of an animal. Well, what we realized is that there are these microRNAs that actually have sequence homology at the end of the CAMP kinase 2 mRNA, which I'm showing here. And what we found is that a mutation in one of the components of this complex, and actually more of them, that's involved in regulating, using RNAs to regulate translation of other RNAs or destroy them, is required to control the expression of CAMP kinase 2 at the synapses in the brain. So here's a normal brain. These structures I was showing you where the fluorescent protein is made. And if we take away one of the components of this complex, you can see how the fluorescence increases here in this kind of punctate way. That's the synapses. And so we take away this protein, this Armitage mutant, and now that inactivates this microRNA pathway, and now this RNA, of course this is the fluorescent protein in this case, but it has this tail on it, and it gets synthesized. Well, it turns out what these animals are that don't have this surveillance mechanism are specifically deficient in long-term memory. They have normal short-term memory, but if you knock out that RNA interference pathway, which is sort of, it's an endogenous pathway, the microRNA, for controlling their expression of CAMP kinase 2 at the synapses, then here's an example here. Just it's Armitage is the name of this mutant, and it's homozygous for the mutation, basically. It has pretty normal short-term memory. This is the performance index in the olfactory test. But the long-term memory relative to a heterozygote, so which is like this, is way down. And so we now are pretty certain that not only does this RNA surveillance mechanism, the microRNA pathway, regulate the synthesis of the CAMK2 protein, but now we know it's occurring at synapses and required for them to form specifically a long-term memory. And this gets even a little more complicated because we know when we activate the cells that there's a mechanism that destroys the protein. So here's the protein on what's called a Western blot. How many people know what a Western blot is? Some people, yeah. And you take a brain in culture, you just take the animal's brain, put it in some culture, and add nicotine, which activates their nicotinic acetylcholine receptors. And the ARMY protein just zap, it's gone. And it's destroyed by this protein machine called a proteasome, which just takes a protein and it chops it up into little bits. So you put in nicotine, all this protein goes into the proteasome, the microRNA pathway is inactivated, the CAMP kinase then gets synthesized. And all of this occurs at the synapses. So we now have this cute little pathway here involving odor and electric shock coming in and affecting this machine called the proteasome, which degrades the Armitage protein, and when you take that away, now you can make the CAMP kinase 2. And I haven't mentioned this or any of that, but that allows the mRNA here at the synapse to get turned into that CAMP kinase 2 protein. And so it looks like it's really a new understanding of one of the mechanisms required for long-term memory. And it all, being found out in a fruit fly, but I wouldn't be at all surprised if it's true. And well, I'll take bets later. But anyway, so that's all I have to say.